We can get started. So thanks everyone for attending this IAS IMS public lecture series. So it's my great pleasure and honor to introduce Professor Gunnar Carlsson. So Gunnar is from Stanford University, and he works on algebraic topology and homotopy theory. And then he switched to the application side. He's actually one of the pioneers in applied algebraic topology, and he is widely regarded as a founder of the area of topological data analysis. So he has published a lot of papers, more than 200 papers with 14,000 citations, and he has also founded 2 companies. And his work has attracted enormous attention worldwide. I personally really enjoy a lot of his work, and it's really benefit a lot from his talks and his papers. And to summarize, I think he's really the world leader that helped to bridge topology and data. So with that, let's welcome Gunnar to give the talk. Gunnar, please go ahead.
Okay. Well, thank you very much, Kellen, and everyone for both for the invitation and for your attending tonight. I'm really happy to be able to give this talk. What I want to talk about today is the idea of trying to leverage topology to try to get some understanding as to what's going on inside deep learning, and hopefully get some understanding which can actually be applied and made useful. So, okay, let's see. Oops. Okay, there. So, first of all, what is deep learning? Deep learning is a methodology based on neural networks. There are a lot of papers that show, that it produces outstanding classification results for various complex data types, images, video, text, molecules also, which is another complex data type. However, there are a few problems with it and then people kind of understand. There are adversarial examples. So, for example, one of the autonomous vehicle companies developed AI. They obviously are developing AI, and they had done so in particular for reading speed limit signs. And it turns out that if you change the speed limit sign a little bit, so one that reads 35, and changing the central bar a little bit so that it's a little longer, so we would still see it as a 3, but the AI saw it as an 8, and that's obviously a severe problem for any autonomous vehicle. There's a general lack of transparency. It's kind of considered mysterious. It's felt that it's mysterious, and so the idea is that one needs to impose some kind of ability to understand what's going on inside it. And this limits usefulness in many key domains. So, basically heavily regulated industries like finance and healthcare. And also we'd like to learn more complex models. Now there's a lot being done in that direction. So, but I think topological models would be very interesting to develop here, or to allow them to create them, because people can interact with topological models. And so what are neural networks? Neural networks, they're the basis for the deep learning, and they are based on the idea that, a learning device should somehow learn or function the way the human or a mammalian brain does, where you have neurons and you have connections between neurons, and one neuron firing affects the other neurons around it. And so the way that's formalized here or one way that it's formalized is in terms of what I would call, a feed-forward neural network. And this is called a completely connected or a fully connected neural network. The idea is that you build a directed graph, which is decomposed into layers here. And in the simplest form, one connects, the layers are ordered, I should say, and one connects any node with any other node in the following layer. So that's kind of what you see here. These are the 2 hidden layers in the middle, and then the output and the input layer. So, and just to summarize how it works, you're given a data set and an output function, and maybe a Boolean valued one, and weights are assigned to the directed edges of the network. So all of these, edges here, all the edges are assigned numbers. which are called weights. So there's a large number of them. What I'm showing here is a very small network. The networks are typically much bigger than this. The activation at a node is computed using sort of a uniform symmetric function of the activations of the nodes connected to it. And so the network is trained by optimization algorithms acting on a set of weights. So the final output then becomes a formula of sorts, but very, very large. And it's determined, its coefficients, if you like, are all these weights, which describe all these different connections between edges in the graph. Now, so what I described was sort of the simplest version there, which was fully connected. And now there's a variant on this, which is what's required to get very good results on images and even text and time series. So these are called convolutional neural networks, and there are certain restrictions that one imposes on the neural networks. So first of all, each of the layers. So you can see here, I've got a layer labeled convolution, a layer labeled labeled input, one labeled feature maps. There are 4 different ones, and then an output. What this says is that we've taken each of the layers and we've decomposed it into grids, which are themselves like the image the input that we have. And so, each layer now looks like a bunch of images instead. And what's done here is you restrict your connections. So the connections are not every node in layer 1 connects with every node in layer 2, but rather only to the ones that belong to points that are nearby in the metric, if you like, on the grid space. So there's this locality of connection. So when I'm in this, when I'm in the upper left-hand corner of the input, it will feed only into the upper left-hand corner of the feature maps at the next layer, and that's called convolutional neural network. That restriction is very useful, because what it says is that you're trying to learn only local features. That is to say, you don't want to have a situation where you're computing something differently in one area of the image from the other. If you're trying to locate a cat or find a cat, you'd want that to be detected in the same way if the cat's in the upper left or lower right. Another point is that you also restrict the weight space in another way, which is kind of a homogeneous way, which says that if I take a square or a little pixel patch in the input layer, and if I move it, then that motion doesn't alter the weights. So in other words, for each of these images or grids, if you like, that are in the first feature map layer, for each of those, have that kind of equivariance property. That is to say, it says that if I'm in the upper left or I'm in the lower right, the weights are still the same. Now, as I move from different grids, you notice there are 4 grids in that feature map space, they can then change between those, but within the grid, it's got that kind of transformation property. Okay, so and that's turned out to be just fundamentally very, very useful for images. So question is, does learning by CNNs, convolutional neural networks, behave like human learning? That's a fundamental question here. It's something I think people believe. This is joint work with my student Richard Gabrielsen. So the question is, what do we want to know? Can we see similarities to what we've found in topological data analysis of image patches? And I'll come to that very shortly. I'll tell you what that analysis showed. Okay. What happens as the network learns? That's a question that one could ask. And what are the responsibilities of the various layers? In other words, do all the layers compute the same thing, or are they assigned different responsibilities? And so, let me see here. Hang on, did we? Yeah, that's right. So here's a description now of some work that we did actually quite some time ago now, some 15 years ago, and it was analysis of a data set of 3x3 image patches in black and white natural images. So we studied only the high variance patches, that is to say, the ones for which there's some significant change within within the patch. So one of the things is that in these image patch data sets, if you don't study only the high variance ones, you will have mostly constant patches. And if you have mostly constant patches, that will sort of dominate the statistics, and it's not very interesting. And so what we'll do here is we'll restrict ourselves to these high variance patches. The second thing is that because the data sets are very, very large, this one is coming from a group, David Mumford's group at Brown, you know, that he had at that time. The point is, in here, that data set is big enough that, in fact, you would capture basically everything. All kinds of patches would come in there. Any patch that you write in this any 3x3 patch, which would be a 9 vector, is probably close to something coming from that data set. And so, of course, that won't be very interesting either. We'll find maybe that our data set, it's been normalized in such a way that it sits on a 7 sphere, the 7-dimensional sphere inside that 9-dimensional space. You know, if we find that it sits on the 7th sphere, that's not an interesting finding. So the idea is to study only the densest such patches, the most frequently occurring motifs. And we'll have density proxies of varying locality. So think here of a kernel density estimator where there's a variance or a sigma factor in there that tells you, you know, do you estimate density only nearby, or do you have a broader definition of it? So we're motivated, this was motivated by the goal of understanding how tuning of neurons in the visual cortex is affected by the statistics of natural images. And so here was the first finding that we had. So, and this was done using persistent homology. I'll talk a little bit more about that later, but the finding was motivated by that. What we found here, though, was initially that the highest density high variance patches were the ones that are shown here. So you can see this is kind of a black to white linear gradient, a discretized version of such, but coming in at different angles. And so the angle is then the circle. So the space of these things is a circle or loopy thing. Now, this is interesting, but maybe not entirely surprising, because what this shows is that these are basically the linear gradients. Well, you know, next to the constant things, the linear ones are the first things that you might expect to come up. Now, what is true is the density varies quite a bit here. So it turns out that the vertical and horizontal patches have much higher density than the 45-degree angle ones. And that's reflected in the next finding, because when we took a more local density measure, so we changed the density measure and took a density measure that, you know, counted only what happened in very nearby points. What we found instead was that there was actually a model, a 3-circle model here. So there's the primary circle, which is the one that we just saw, and that's in that upper middle, and then 2 secondary circles, one vertical and one horizontal. And what you'll notice is that those two patches, those 2 circles, each intersect the primary circle in 2. You can see there that 2 red ones on the lower secondary correspond to the 2 red ones in the primary. And similarly, the 2 green ones in the secondary, the other secondary, correspond to the 2 green ones in the primary. The 2 secondary circles don't intersect at all each other. And so this thing is actually something that came out, and this is much more surprising to us, actually. You can see what it's saying. It's saying, look, there are edges that are gradients, but now there are also lines in the secondary, the vertical and horizontal secondary direction. So a line here means, you know, a black line in the middle of a white region, as you might see at the top of the secondary circle on the right. Now you could ask them what happens if you relax the density threshold a bit more, and so that you can actually include some of those 45-degree patches as secondary circles. And it turns out that the model then is this gadget here, which is a Klein model. So the thing you have to remember about this is that you've got connections made. So if you look in the upper left-hand corner and the lower right-hand corner, what you can see is that those 2 are identified. And also if you look, say, in the middle, and let me see if I can get to this. Yeah, this one right here is connected with this one. And this one is connected with this one. So there's a twist going on in the identification, which is interesting. And then secondarily, there's this one is connected to this one, and this one is connected to this one. This is an interesting object. It doesn't actually fit in 3-dimensional space. You have to, if you try to do it with a piece of paper, you know, make all these identifications, you'll find that you'll end up tearing the piece of paper. But it is a mathematical object that exists in its own right, and we'll come back to it here in a bit. So let's just come now to the motivation for this, which was to study questions of the, the neuroscience of the visual pathway, the visual, the visual pathways. So the primary visual cortex is the lowest level processing unit beyond the retina. So the higher levels, and they have various names, there's more of them than these, but V2, V4, and the lateral nucleus or lateral nexus perform more abstract tasks. So that is an observed fact in the mammalian visual system. And Hubel and Wiesel many years ago showed that there are individual neurons that detect both edges, which go between a single edge between a black and a white region, and also other neurons that detect a white line in the middle of a black region or a black line in the middle of a white region. And this is consistent with the idea of compression of frequent signals. So if you think about what we found there, we found that, well, the lines and the edges are the most frequently occurring things. And then this model, this finding of Hubel and Wiesel is sort of consistent with that, and that the visual cortex has adapted itself to those frequently occurring signals by invoking the idea that a single neuron is devoted to detecting them, because a single, you would want to detect things that occur frequently, you would want to detect them with a single neuron. And so, and here's sort of a picture of the situation. Okay. And you can see here on the right that more complex objects get understood or processed at sort of higher levels. Okay, so now what I want to do, we're going to try to study what happens with neural networks, with convolutional neural networks. And so in order to do that, the analysis we're going to do is topological data analysis. And the question that we're asking, remember, is does a neural network behave in somehow the same way as as the human or the mammalian system? So let me tell you about topological data analysis. So data has shape, and the shape matters is our mantra, so to speak. And so let me remind you how we think of data. So if I see this data set right here, I would think this looks like a line. This looks like something where linear regression works very well, and indeed it does. And linear regression is a well-established method in mathematics, statistics, and computer science. And it is approximating data sets by lines or planes and so on, linear regression. However, there are data sets for which it's not appropriate. So this is a data set here, which the salient feature here is that it breaks into 3 groups. And you can think of that as also being an approximation problem. This is, cluster analysis, which is the approximation of a data, set by a discrete geometry instead. So you might think of those 3 points, you might not think of them as a shape, but they are also a shape. And cluster analysis is the idea of approximating by such things instead of approximating by lines. But now as we study more and more data sets, we might run into one that looks like this. And so this is an interesting one. You'll notice it has loopy behavior. There's no line that's going to capture this. And there's also no clustering that's going to capture this. And so we'll say here, okay. There's a loop. And we might say, oh, as mathematicians, we might say, great, let's build a loop detector. That'll be our third part of our arsenal, if you like, of methods to approach the data with. But at this point, I'm possibly going to worry a little bit, because I'm going to say, do I have confidence that this loop is sort of going to be the last thing, or that there even is going to be any kind of finite list of shapes so that I can build special purpose algorithms for each of those types of data sets, or shapes of data sets? So, and frankly, I guess I'm not convinced. One isn't really convinced that that will happen. And especially when confronted with a data set that looks like this one, which is the letter Y. Now, this one might come up, for example, in data collected from sensors aboard an airliner or an airplane, where you might have a central area in the middle there, which would be flying at altitude in non-turbulent conditions. But then I might also have the exceptional parts, which are the tips here, that would be, say, takeoff, landing, and flying at altitude in turbulent conditions. And so now this one is a letter Y, or I'll call it a flared thing. It's clear now that this is another shape this comes up rather frequently, and that one doesn't really want to believe that we're going to be able to capture every possible shape. Instead, we should develop a methodology that can handle any shape. And that's what topological data analysis is. So, and just to give you a sense of it, here the Königsberg bridge problem was something that Leonhard Euler worked on. The question that he posed was, can you cross all the 7 bridges across the River Pregel in Königsberg? Can you cross them all without crossing any bridge twice? And in this situation, there's lots of information. There's the size of the islands and the depth of the river and the length of the bridges and so on, none of which matters to the problem. Euler identified then the core of the problem is really this network model on the left here, which is where the nodes labeled A and D are the islands, and this B and C are the riverbanks, and then the bridges between them occur as you see. So what I want to do now is I want to do something similar for all data sets that can work for any kind of data. And so what we will say here is first that we will assume that we have a data set. So you can see over there, I've taken a very simple loopy data set, and we have a projection of that onto the real line, or frankly onto the plane, or even onto space. But I want to say here, we've done this with the y coordinate function here, but in general, we won't be using a particular coordinate function on the data. We'll instead be using something statistically meaningful, either like a density measure, or a centrality measure, or perhaps some machine learning coordinates, say a PCA1 and PCA2. But the idea then is to take this line or the plane and divide it up into overlapping bins. So we cluster, those bins, we'll take the inverse image of those and find bins in the data. So you can see it looks like this. See, I've divided up the line into overlapping intervals. And so you can see that in the data set here, they overlap. You'll notice at the top, there's orange, which is the same as the orange in the next layer down, and so on. And at this point, then what I say is now I'm going to cluster each of those bins. I'm not clustering the whole data set. I'm clustering simply the bins. And so when I do that, I will get 6 clusters, as you can see, because on the left there, I've got these 2 clusters here. Here, there's only 1 cluster. Here, there's 2 clusters, and again, 1. So I've created these 6 clusters. The thing to keep in mind here is this is a soft clustering in the sense that the clusters actually overlap. It is not a hard clustering. And that's an advantage for us, because what we can now do is we can say, well, now we have a bit of a proxy for density or closeness, because what we can say is that 2 nodes are connected if they share 1 or more data points. So if I do that here, you can see now I have something which is a hexagon, which is a rough coarse model for for the circle. We've lost some curvature aspects, of course, but nevertheless, it is a good representation of that data set. Now, the situation is not this. I've of course shown you the most optimistic situation. So it's not always that we get a perfectly accurate description of the data set, but what we always get is some kind of image, some kind of idea, some kind of map of the data set, which often can give you an idea of the taxonomy of data points. So let me show you here. So for example, there's a group at Mount Sinai working with Joel Dudley at the Mount Sinai Hospital that studied a data set of about 11,000 type 2 diabetics. And it was, it was some genomic data, and it was also health record data. And what they found was that this was the graphical model of those type 2 diabetics. Now, type 2 diabetes had been thought of as a single disease, but in fact, what this shows, and what they found, is that it's really 3 separate diseases, and they have separate properties. So they have one of them is very much connected with cancer. Another is connected with neuropathy. And they don't overlap. This is very important from the point of view of precision medicine, because what it can allow you to do is to say, I need to develop a drug, say, I don't need to be able to solve every type 2 diabetics problem. I need to solve one of these groups, and that's an easier thing to do. So in general, the more understanding of the disease that we can get, the better off we are. Here's another one. This is a description from the David Schneider group at Stanford. He was studying the progression of disease, of infectious disease. So if you think about this, when you're studying infectious disease, you have a data point, and I should say he's measuring physiological things and genomics. So you start out at a certain point, and then as you get infected, your state starts to change. And you move along an arc until at a certain point the immune system kicks in, and then you start to return along a different arc back to the first part of the or back to your initial point. And so in this case, because of the fact that this, you start out healthy and you return to the healthy state, an appropriate model here is a loopy one. And what he found is, yes, he found loopy models in all these situations. You can see mouse malaria, human malaria, and so on. Microbiome kind of data as well. And what he was able to do from this was to find good predictors for who's going to actually survive, who's going to make it through. That's an interesting thing. But also, furthermore, a better understanding of the actual state that you go through, because you don't want to encode this just with time, because not all patients or not all subjects go through at the same time, at the same speed. In fact, it can vary, stay at some parts longer and go faster through others. But here you can identify the state in a time invariant fashion. This was an analysis that we did, a microarray analysis of breast cancer. This is a data set of 272 patients, 1,500 high varying genes. And what we found here was a group labeled CMIB tumors down there, a specific group of tumors with a particular genetic expression profile. Is about 8% of the patients, and they had perfect survival. On the other hand, that upper thing there is it includes the triple negative breast cancers and is a very severe outcome. We'll return to this data set in a bit. But here's an interesting feature. So this is breast cancer, but this represents two different breast cancer data sets. And the idea is that one of them is the top two are the NKI, which was the data set that we just showed you. The other one is done using a whole different platform, so a different measuring methodology. So the measuring methodologies are entirely distinct. You can't compare them coordinate by coordinate because it's different gene sets and so on that are being used to study it. On the other hand, though, what you see is that even though the measurement mechanism is different, nevertheless, the structure of the data sets is quite similar, because you can see both of them have that large, a large structure called the high ESR1 levels, and then they have some clusters up above them. And the clusters are those are the non-survivors, that the survivors are the part in this bigger structure. And even there's a lot of consistency that one can see. So the idea of the topological analysis is that it can measure things in such a way that you can get away from having to compare coordinate by coordinate. Another one here for us that was kind of interesting was we had a group of collaborators who had developed a predictor for who's going to survive in an intensive care unit. And so here the data was questionnaires filled out by the personnel in the intensive care unit concerning the patients. And so we built a model for these questionnaires. And what you can see is in the upper left, that is where that model is being colored by the predictor value. So what you can see there, where it says predicted survival, the red part on the right is actually quite that's where the prediction is that this person is going to have trouble, this person may not survive. On the other hand, the blue on the left is where you have people who you say, well, these people are probably definitely going to survive. Maybe they even leave the intensive care unit. But now what I can do is I can color instead, not by the predicted survival, but I can color instead by the presence of actual survivors. And so, or actual people actually dying. So you can see there's a lot of consistency in what happens on the right there. But what you can also notice is that in the upper left, and I'm looking now at this one right here, this little group right up here, this is bright red, whereas up here it's bright green. So the predictor is actually not performing well here at all. And so one wants to know that. And when, as you dig into it, what you find is that this is a situation where some of the questions were not answered. So these people in this group were ones for which two particular questions concerning the mood of the patient and so on were not answered. And it turns out that those were an important feature in trying to predict what's going on. So now, of course, you could take two separate approaches. You could build a new, better model that includes those things, or you could just say that, well, we're not going to take this model too seriously if those questions aren't answered. Here's another one. This is a collection of sequence data. It's coordinatized with a copy number variation, if that's a numerical quantities that you can compute based on sequences. And it's from three separate populations: a Chinese population, a Nigerian population, and a US population. And now dividing up the groups is something that anybody that calls himself a method for data analysis can do very readily. They can break it up. That's not a hard thing to do. But what you'll notice here is, and so in fact the red one is the Chinese group, and the yellow I think is the US, and the blue is the Nigerian. But what you'll notice is that the texture is a little different. So if you look at that red data set on the left, the Chinese one, you can see that it's more carpet-like, whereas the other two are stellated. They have a higher degree. And it turns out, you might think this has something to do with the biology, but in fact, it has to do with how the data was handled. So it turns out that in the Chinese study, they had taken the data processing step of removing any parent-child pairs. So in other words, if the parent was in there, they would remove the child, or if the child was in there, they would remove the parent, whereas that step wasn't taken. We always like to think that we're dealing with the data, the science, but in fact, we have to be cognizant of the fact that, the data processing, the way it's handled, is always a key feature. And here's a similar one. This was a collaborator of ours that came in. He was studying cell types, I believe it was, and he saw this cluster in the upper left there, which was a red group. He thought he had found something extremely interesting, that it would be his next, publication in a high-profile journal. But in fact, it turned out it was mislabeled data. So, you can even find examples of working with, the data processing side of things here. You can take the raw data and study it this way. And now here, this one is not biological. This is a data set from the World Values Survey. So World Values Survey is a survey which gets taken, every few years. It's 250 questions. I decided to sort of look at it for the US respondents, of which there were 2,000 or so, and to study only those questions which concerned the trust in institutions. So the questions were, how much trust do you have in the federal government in the US, for example? How much trust do you have in the press? How much trust do you have in the church, and so on? And so this is the overall model. And so there were only about, I think, 15 questions of that order there. So there's another question which was not used in building the model, but which asked, where do you fit on the liberal to conservative spectrum, if you like, from left to right? And so what we found then is that based on the trust in institutions, you could really identify pretty well the answer to that other question, the liberal versus conservative. And you can see it's colored red for conservative and blue for liberal. Another one says, well, since we're talking about trust in institutions, I could ask about the aggregated trust in institutions. In other words, how do I trust in all institutions? And so that's a feature that I could compute, and that turns out to be interesting as well. And it shows up here that, that on the upper right there are people who have no trust in any institutions. In the lower left, they have a trust in a lot of institutions. And so, for want of a better word, we call them non-establishment and establishment. And another question then that was in there was, do you think you have control of your life? And so what you find here is that this question also is correlated with this picture in a way that we might not have understood before. And so you can see that the lack of control in the life is actually in that upper part right up here. And so that lies right in between. That is sort of people who are, you know, are liberal, are on the left, and non-establishment. So they are the ones that maybe lack the most, or feel that they lack the most control over their lives. This was a question about, you know, basically an immigration question. You know, should the government in your country, should it favor people who are already in the country versus people who are immigrating? You know, obviously a controversial question. And you can see here that this question is modeled with the disagreeing with that on the left and agreement on the right. And a related question here was, do you or do you not support the UN? And notice it's the United Nations. It's very correlated with that previous question. Okay. So anyway, summary, topological modeling. The model output is a network, not equation. So we might think about mathematical models as being equations. I know I generally have thought of them in those terms, but in fact, maybe a network is a useful model because it's going to have a lot of functionality, it turns out. And so first of all, it is a visualization so that you can look and get an overview of your data, but you can also perform selections of subgroups this way, because if you've got a network layout where each node is a collection of data points, I can, you know, lasso it if you like, and then get a group of nodes, which would give me a group of data points. I can do segmentation, graph segmentation, to produce a taxonomy. I can do coloring by various variables. So just like we saw in the you know, in the World Values Survey, I colored by other questions to kind of see how they worked. And this gives you also hotspot analysis. Like we had in that ICU, intensive care unit, there was a sort of that small red hotspot of death up in the upper left. And one can explain the difference between groups and justify them and do so-called topological feature creation. We'll talk a little bit about that, model assessment and improvement. Okay. Now, so here, let me now show you. So I've just shown you a lot of the ways to use this mapper description, mapper methodology on a lot of different data sets. What we're doing here is we're using it now on the weight vectors that define the neural network. So remember, in the neural network, what you've got is you've got a locality of connection. So if I have 9, you know, if I have a data point, or I should say a pixel, it gets connected only to its 9 corresponding neighbors, you know, it point, and then everyone that is, you know, one step away from it. And so it's a 9 vector, just like we had a 9 vector in 3x3 image patches, process these weight vectors in exactly the same way as we processed those image patches, and now computed, you know, developed a mapper model for it. And you can see here that we've got basically that same primary circle that we saw before in the image patch analysis, but now it's being reflected in these weight vectors. And, you know, you might think, you might be a little skeptical. You might say, well, you know, didn't you just kind of, I mean, there's lots of parameters in this modeling mechanism. Maybe you fudged it to make it be like that. But then we have persistent homology coming to the rescue here, because persistent homology is a mechanism that sort of tells you whether you have loops in a data set or even higher dimensional geometric features. And so here, the dimension 0 says that it's connected, but then the fact that there's this long bar in the barcode says that there really is a loop in this data set. And so we really, it really is a loopy thing. Now, one of the other questions that we asked was, well, what happens as you learn? And so this is an analysis now. What the previous one, let me say, the previous one was MNIST. This was on a data set of hand-drawn digits. It's one of the standard, you know, benchmark data sets in this area. Here's a more complicated data set, which we put in. And I'm just going to show you that one can get an analysis of what happens in this, in this kind of analysis. So these are all mapper pictures of the weight vectors that come out. And you'll notice that what's happening on the left here, it's kind of carpet-like. And let's look at layer 1 first. So what you see there is it's kind of random, but as the learning process proceeds, so you look at the bottom here, there are various iterations in the optimization scheme that ultimately results in the neural network model. And you can see that what's happening is it's starting to concentrate around the edge. And in fact, then after 400 iterations, you've actually got that loopy behavior. This is actually that same primary circle that we saw. And by the way, it's colored by a number of points in here. So when you see this one here, there's many more points in each node here than I have in the middle. And so this kind of looks like a circle as well, as does this. But what you'll notice is happening as I move from 400 to 500 and then to 900, the circle is degrading. And the circle now gets to where it's something concentrated in the middle. And that turns out to reflect something that we can think of as a bullseye. And then I've got these 4 corner points. So that's the first layer. Now the second layer, you can see, it's random, and it kind of stays random for a while, for 100, 200, even 400 iterations. But now at 500 and 900 iterations, you can see that what's happening is that as that primary circle in the first layer is degrading, you're starting to see some concentration around the boundary in the second layer. And that sort of continues until the end when it's actually pretty well defined. So you can see that in this case, what's happened in this neural network is actually that what you've got is you've got the first layer that's capturing a bullseye, and then certain of the primary circle features, and then the primary circle is really being captured on the second one. We don't believe that's what happens in the human system or the mammalian system. We kind of believe that this is what happens because of the fact that the network is so shallow. There are only 2 layers, and it's a complex data set. So, and what I'll show you here is in a minute is an example that we think is more representative of what happens. But just to give you a sense that we actually found this 3-circle model in here, it turns out that the dimension 1 for the 3-circle model is supposed to contain 5 bars, and that's confirmation. This is happening in this CFAR 10 as well with a looser density thresholding. And now here, this is an analysis of weight space in a VGG16, which is a much deeper, more complex network that's trained on ImageNet. And here you can see what's happening. Here are the findings that you can see the primary circle occurs in both layers 1 and 2, and then you start to get some secondary circle information in level 3 and 4, it turns out. In level 5, you're actually getting something which is not included in that Klein model at all. This is a crossing instead. And you can see a similar crossing and bullseyes in layer 7, layer 10, layer 11, and 12, or 11, not so much in 12. So this is kind of indicating in VGG16, the responsibilities are that, 1, 2, well, first of all, everybody has responsibility for the primary circle, but then some of the higher ones have specific responsibilities around crossings and bullseyes, which are things that one might think of as you get into kind of second order things. So one thing that one could do then is you could hard code the primary circle and Klein model into features. So you would say that the features coming out of these things, should be given to the network right away at the beginning. And in fact, what you do that, and it allows both for speed up and generalization. So it's a speed up of, a factor of 2 for MNIST, which is an easy data set, and a factor of 3.5 for SVHN, which is a house numbers data set, much more complicated. Also, it would allow us to generalize, to train on MNIST, and to evaluate on a house numbers data set, and to get some sort of accuracy there. So let me explain one thing that, again, MNIST is an easy digit data set. SVHN is a hard digit data set. So you can imagine trying an experiment where you train a convolutional neural net on MNIST and evaluate it on SVHN, house numbers. And when you do that exercise, you get 10% accuracy. So in other words, it's effectively random because this is a 10-way classification problem, there being 10 digits. When you instead, feed it the Klein-Bottle features, the generalization improves from 10% to 22%. The accuracy goes to 22%. Obviously not where you want to be, and we'll do a little better in some analysis that we do going forward, but it shows the direction that you can, notice that you can improve generalization this way. Okay, so now here one can build graph neural networks, as they're called. So the convolutional situation uses the grid structure on the set of pixels in a critical way. So pixels are the features for image data sets. We use this a priori geometry and use it in designing the neural net and in doing the image, the feature generation or the feature engineering. And it's important in restricting the class of formulas that are considered. So here, but we can ask the following question. In this case, we had done a scientific study. Well, let's say, for convolutional neural nets, you have an a priori geometry on your set of features, on the set of pixels, namely it's a grid geometry. But what if there is no such a priori geometry? So, well, you might ask, is there a discovered geometry? And we've discovered feature sets that carry the geometry. And so those are spaces that are primary circle and Klein-Bottle and so on. Each of those things generates a feature by taking dot products against a particular patch. So you can use the geometry to do feature engineering and to design architectures. So now let me talk a little bit about feature space modeling, because this is how to take this and then say, I don't necessarily want to do a science experiment, the kind that we did here with the Klein-Bottle, or do a scientific study. Is there a way that I can build some kind of method for understanding the space of features that are being used? And the answer is yes. You can get given a data matrix. You can also consider its transpose matrix. So that's taking the columns, which are features, and making them rows. And so what I can then do is I can build a model on that, a mapper model on that. So this compresses and recognizes correlations among features, and each row of the original matrix now gives a function on that data set, on that feature set, and it turns out on the nodes of a topological model. So what I can now do is I can study individual data points or groups of data points by evaluating them on the topological model and representing that through coloring. So let me remind you here of the same analysis that we did. This is the analysis on the patients here. This is a model for the 272 patients, but we've got these separate cohorts. This cohort A, cohort B, poor survival here, perfect survival here, and middling survival here. Now the question is, how are these being represented genetically? Well, so what I'm showing you here now is a geometric model or a topological model, a mapper model of the genes in this study. So those are the columns. And so now I can color by that cohort A, which is the perfect survival one. And you can see that it goes from dark blue here at the bottom up to bright red at the top. Whereas the poor survival is bright red at the bottom and blue up here, although there are some genes that apparently never change at all. And then cohort C looks pretty similar to cohort B, except it's just kind of weaker coloring and smaller coloring here. But it's not very much like cohort A. So it sort of suggests to us that cohort A is something special, and the other two are kind of the normal run of things that you can see being just a weaker or a stronger version. Another thing that we did here was we obtained some gut biome data, as it's called. So this is bacterial populations in the human gut. And so the features here are particularly correspond to bacterial subpopulations, and you're measuring abundances of them. And so notice again, the features are the same for every one of them. The graphs are the same, but the healthy ones are the ones in the lower left here. So this is the coloring by the healthy group of patients. This is the coloring by those having ulcerative colitis. You can see it's quite similar to the healthy ones, except there's this very bright red bit here that's not seen here. So it tells you there's a kind of a single group of features that are actually capturing, ulcerative colitis. Similarly up here with Crohn's, you can see it too also looks quite a bit like the healthy one, except it's missing this bright red one right here. So this becomes quite a powerful way of even doing by hand analysis, because it identifies groups of features for you very quickly. And it is possible to kind of change resolution on these things and really find good ways of trying to understand what's happening in your data. And let me just, this is just a formalism for generalized convolutional neural nets, but a particular version of that might look like this, or have components that look like this, where we have a Klein model and I make connections like this. Just like before I had a grid, I will make connections that are local in the Klein model geometry. So, and I now want to sort of conclude with, some experiments with some of these topological neural nets, and really it's topological, feature engineering. And this is joint work with Effie Love, Vasileios Maroulas, and also, I'm sorry he's not in here, Ben Filipenko. So we were talking about generalization before. And so one way to test generalization would be to take a fixed data set like MNIST and noise it up a little bit. So add Gaussian noise to all the features. And so then what you can do is you can train on the noisy and evaluate on the on the clean, and also you can train on the clean and evaluate on the noisy. And so if you look here now, what you can see is that when you do this, when you perform this noising it up, this right here is an example of a traditional neural net. And so is this one right here. So the performance on accuracy, as you can see, is not very good. It's under 20%. Okay, but when you add these, so all these other ones are various variants of our thing here. So, it involves the primary circle. It also can involve the Klein features. These are the ones that involve the Klein features. And it's two different architectures corresponding to those. And so what you can see happening here is not only do you learn quite rapidly and you get up to a reasonable degree of accuracy, or 0.8%, and then it tapers off. That's something that one understands happens a lot in neural nets, because that the accuracy after a certain point actually tapers off, because there's a factor built in for cross-validation there so that sort of test accuracy is what's being measured, not sort of absolute accuracy. But anyway, the main point is that you get to a very good point very quickly by comparison with the standard neural nets. And here, this is the similar analysis here where, where we trained on the clean. And once again, the orange and green, which are the Klein feature ones, do the best. Here are the primary circle ones, and then the ordinary neural net. Similarly, if you remember, we talked about the generalization from one data set to another. And so here, this is again SVHN to MNIST, and MNIST to SVHN. And these two down here are the ordinary neural nets. These up here are the Klein features, several different formulations of those, variants of those. And you can see here you get to nearly 50%. SVHN here is the harder data set, so that's to be expected. This is MNIST to SVHN. Here we're getting, well, it looks like we're getting to 25, but in fact we actually got up to 35. So there was a significant improvement over the previous analysis. And I won't talk so much about that one. The learning rates or learning speed here, you can see again the speed in particular for SVHN, which is the hard data set. These are the Klein features here. These are the circle features, and then here is the normal one. MNIST is a simple data set, so there is improvement, but it's at the early stages, and ultimately you get you arrive at the same point. What's interesting here is that this USPS is a data set of smaller images. It's not 28 by 28 as the other ones were. It's 16 by 16, and smaller data set. And so in this case, we still do better with the Klein features, but the ordinary neural net, for reasons we don't understand, outperforms just the straight up circle ones. So somehow the circle ones are not captured so well in that data set. But it means that lines are important in that one. Okay. And now this one to me is perhaps the most interesting. So this is video. Now with video, the features that you want to generate will no longer be, on a Klein bottle per se. I mean, because after all, there's time variation going on. What's interesting is that mathematically, as you reason about this and you reason about the Klein bottle model, you can see that a reasonable model for the video would be the so-called tangent bundle to the Klein bottle. Because the tangent bundle represents infinitesimal transitions, within that image patch space. So, and I should say, we didn't do any statistical study on that. Just the fact, the understanding that we had of the static case allowed us to make a good guess here as to what would work well for video. And indeed, we found that it worked very, very well. So here, because it's video, it needs much more complex networks. It's not these little 2-layer things. It's a 12-layer ResNet with, with the Klein features or the tangent Klein features plugged in. But what you can see happen here, this right here is a standard neural net, plugged in, and this is the Klein thing. And so you can see that you get much better results. You get to a better place, and you get there much more quickly. And I should say, this is a classification problem from the University of Central Florida video classification data set, which has various human, classes of motions. And so that was quite interesting to us to see that this could be done. People work on this data set, people who work on it generally develop, they calculate features, they do feature engineering from a vast array of video data, from around the web. We trained, on the other hand, just on the data that we had within this data set. In other words, no a priori feature generation coming from statistics of images coming out. And the reason that you would want to do that is you may have many situations where you're say in medical imaging, for example, where you wouldn't want to train on generic images. You'd want to train on things that are like what you have, and in particular, you'd like to be able to reason about it and be able to adjust the feature engineering so as to get good answers. Okay, so, and let me just finish then by saying, here's a summary. Deep learning is data hungry. Deep learning typically uses a lot of data. It has trouble working with prior knowledge. It has trouble with generalization and adversarial examples, and it cannot be engineered. By the way, I'm quoting here, this really comes from a paper by Gary Marcus, who studied a sort of a friendly critique of deep learning, and these are the points that he came to. And what we believe is that what we're doing here is allowing you to do some of all of this. In other words, it's a step in the direction of trying to resolve these problems. So we're very happy with this, and we believe that what's going to be very interesting for us is to study, say, the mapper models for the feature space and do feature engineering with them. And with that, I will thank you for your attention and happy to answer any questions.
Yeah, thank you. Any questions? You can just unmute and ask that.
Hi,
Yeah.
I have a question.
Yeah.
So the visualization, right, is on the data set, is it? How do you actually do that again?
How do we do that? Oh, so that was, well, let me go back. Let's see here if I can. That was the mapper thing. So the way that the visualizations come, let's see. Yeah. Sorry, it takes so long here. Let me get there. It was this, it's this construction. Oh, sorry. So if you remember this, where we took the bins and we created the bins using a projection, overlapping bins, and then performed a clustering step in between. And then realized that this was a soft clustering. And so we draw edges between any two nodes whose clusters overlap. And so we get a graph out of it. So now the graphs you saw were obviously much more complicated than this one. But that's just well, that's life. This is kind of this is to give you a sense of the construction, and then I showed you lots of examples of them. So everything that I showed you here was an example of this construction in action. Does that make sense?
To clarify, so basically you just you use like a EM type of clustering algorithm, and then apply it to like the weights of the deep learning and all these things, or the data set, right?
Well, except remember, so first the first step is to take this set of weight vectors, and I'm going to create a projection going out. My recollection is that this was a measure of centrality, was the projection that we used. So we can build a map, then we can build this mapper model by clustering the individual bins. Again, it's critical. You never cluster the whole thing. You use the projection to be able to build things that you can then cluster, bins that you can cluster.
Well, so how do you do the bins, like based on the?
Yeah. So let's suppose that we have one projection. Let me show you here. Yeah, so if you see here on that line in the middle, we've actually divided up the into overlapping intervals. So you can kind of see them here with these brackets. So here's an interval, and then here's another interval, and here's another interval, and here's another one, and they overlap. And so we take the inverse image of each of these intervals up here, and that's what you get here. So you have this one, which is the inverse image of this interval. This one, which is the inverse image of this interval. This one, which is the inverse image of this interval. And this one, which is the inverse image of this one. So I have divided up the data set into 4 bins. And now I perform clustering within each of those bins.
Okay, I get it.
Great, okay.
Hi, can I
Hi.
just build on the question just now? So like for this, let's say the plot on the left, do you plot according to the weight of the neural network, or like what are the features of the plot, like before binning?
Oh, well, let's see. So the features would simply be the weights. You see, remember, because of the neural network, the locality of the connections, that the weight vector for any given grid, or any pair of grids, one grid in layer 1 and the grid in layer 2, that creates a weight vector, which is a 9 vector, just like an ordinary image patch would be a 9 vector, right? And so what we then do is we say, well, what are the most free? Well, first of all, we mean center it, so that the weight vector is mean centered. And then we'll take a measure of density on that and compute only the densest ones, the most frequently occurring weight vectors among the things that we had. And by the way, the study was done by repeating the analysis many times. In other words, we would run run the network many different times, and so getting different answers each time. But doing the statistics over time, what we found with this.
Okay, understand. Then just now you showed there's like around 13 layers of neural network. Like it ranges from 1 to 13, and then it gives different visualization, right? So like in that case, how do you know which one is more desirable, or does it? Yeah, does it have any...
Let me see if I understand what you're talking about here. Are you talking about this thing right here? I
think
this one?
A few slides after. Yeah, this one.
This one, yeah. Right. So first of all, I show the visualizations, and what you do then is you then go in and say, well, for each region in that graph, I can select the collection of nodes, and then I'm just putting out a representative sample there. You see, so because these things are now 9 vectors, they look just like image patches, and so that's kind of what what this is representing is a vector. Say if I look at this one, this is representing a weight vector, which is very heavy here in the middle, on that, but then grayer on the sides, and then white, very low at the corners.
So do you mean that based on the result, you sort of see like which one fits the narratives more, like if which one allows you to interpret the data more?
No, it's not. I'm not. I'm not going after any particular narrative. What I'm trying to do instead is to actually say, let's find a way to interrogate meaningfully, what's happening in the weights in each layer. And so we build these models for it, which is something that's done systematically. And then now what I'm able to do is I'm able to say, look, if I have one of these networks, I can select, say, this region here. And I will select and I will look at all the patches that occur in there, or all the weight vectors that occur in there. And what I see is that there's a lot of similarity between them because they have been clustered together. And so that's what this is saying. What this is saying here is that this region right here is one, that for which this is representative, and this one here, this region here is one for which this is representative. But if you like, I mean, I wouldn't call it a narrative, but it's an examination of the data points that are in that very local region of the visualization, and and then finding a representative one. I agree. If in fact they were not very homogeneous, that would be problematic. But it's not problematic because in fact they end up being very close to each other.
Okay. I think I understand. Thank you.
Yeah, thank you.
Any other questions?
Hi. Another. So at the end, right, you were saying that you have better neural networks compared to the normal one. So what is the better neural networks?
Sorry.
Like
So what we're talking about here, I should say the improvement comes mainly from the feature engineering, and the method is that we do, so that what we do is this: we create features as follows. So if it were the Klein bottle, each Klein bottle corresponds to a particular weight vector, and so now I can get a feature on my data set of images, say, by taking any patch in that image and taking its dot product with that patch on the Klein bottle. So each image now gets represented as a function on the Klein bottle, or if it's video, it's a function on the tangent bundle to the Klein bottle. And so it's that, it's the ability to now to produce those features and feed them in a very systematic way that allows for this improvement.
Also, so to clarify, so basically you pre-process it with some feature extraction, then after that apply the same neural network.
Well, yes, except in some cases we've also modified the neural network. Most of the improvement is due to the pre-processing, but we are still working on the network. And I have to be clear that here on the video, we had to impose it's feeding into a ResNet, for example, where you're feeding in this geometry of this graph neural network.
Wow, okay, good. Thank you.
Thank you.
Any other questions? Okay, so Carlsson, maybe I have a question. So for the data, you see the great improvement with the TDA information getting involved. I'm wondering, for what type of problem or data problem do you think that the topology and geometry will contribute more greatly than existing models?
Yeah. So look, I think it's like this. I think topology has a tremendous amount to offer in situations where you have data sets that have a large number of features. Where it's useful to compress them so as to build a topological model which is smaller, and so that you can actually view it as a kind of complicated dimensionality reduction. And so I think that's a very very good situation for it whenever you have a lot of features. But but frankly, I sort of believe that, you know, putting them inside the bigger networks as well, I should say, putting in the graph structures at the higher layers as well is also going to be very useful going forward. You have to, though, allow the network the sufficient freedom to capture what it needs to capture. So yeah.
It seems that for your last example is that you know, when you add in the noise, it gives a lot of trouble to traditional learning, but maybe the topological information is more like resistant to this noise, so it can have better performance.
Maybe so.
This video.
Yeah, I think that's quite possible. You're referring to the video example?
The minced one with the noise you mentioned, yeah.
Oh, yes, exactly. Oh, I see. Yeah, that's right. I think the features are somehow, you know, the fact that we've allowed we've taught it to get to those features early on somehow is very helpful, so that it can train given that it already knows that it's got the features that correspond to the edges that it would otherwise have to learn.
I see. Yeah, thank you. Yeah, any other questions? If not, let's thank Gunnar again. Yeah, thanks for
All right. Thank you so much.
Yeah, thank you.